The next two in the program, or three in the program before lunch, is Jason Clark with South Dakota State, and he's going to be talking about soil health and phosphorus management. So thanks for joining us.
Yep.
Yeah.
All right, well, it's good to be back here in Minnesota. It's been a while since I've presented out here. So to kind of give a perspective of phosphorus and some things that we're working on in South Dakota and working on improving some of our phosphorus recommendations as well as revisiting them. And the first little bit here, we're just going to kind of go over some of the basics so we're all kind of on the same page with some of the nutrient aspects and some of the strategies we're working on these. Because what we typically do with our soil test results, we've taken our soil samples, we've taken the lab, we've gotten our soil test results right here, and then based on those soil test results, we get recommendations. And with those recommendations, kind of there's two pieces to how the lab or how the consultant or whoever gives you those recommendations gets those, what data is needed. And the two basic parts of that data is first the correlation data, and put it simply, this is basically when the soil test level tells you you're not going to respond to fertilization. And the second part of that is our calibration area, which is the rate that you need to apply to get that optimal yield that you're looking for. So these are kind of the two aspects, and to make sure we're all on the same page, we're just going to go over some things here. So here we have our soil test phosphorus level, and depending on where you are in your soil tests, we're not going to go over the ins and outs of each of these, but these are some of the tests that you may run, and depending on what your soil type are. And you can go from a low to a high, and we get different levels. The important thing to note about some of these things is this alone doesn't tell you what to do. It's just information. What really needs to be done with each of these different tests is you need to relate it to something. And usually relate that to going from a high to low as some sort of measurement of yield or relative yield in that field. And once we can do that, then we can create relationships, and then we can make decisions based on when we should not. Something like this is once we get to this level here, we flatten out. So we typically no longer see fertilization responses. So this is one piece. This is the correlation where we're trying to figure out what level this is that we typically don't need to fertilize, and does that change based on our environment, based on our management system, and other things like that. So that's our correlation, which we've been working on in South Dakota. So we'll go over some of that data. The second is our calibration. We still need this, but we also, instead of having yield over here, we're looking at fertilizer rate. So we want to know that relationship between soil test and the fertilizer rate we need to apply in order to get that optimal yield. So those are the two basic pieces, and then kind of a lot of how we've been doing this and how we've been working on it in South Dakota and a lot of other people. Typically, you're looking at small pot trials or large farm trials. You're looking at different rates on that field. This is just an example from 0 to 150 pounds of PTO5 they could be putting out. And then what we do with that data is you're looking at that yield response right here to that phosphorus rate, and you're going to get data. Can various look like something like this. So we have a lower yield, we add more phosphorus, we increase that yield until we hit a certain point. And this is what level of phosphorus fertilization that needed in this field to get that, which is about 71 pounds of phosphorus per acre. So from this field, for an example, this field was at 12 parts per million Olsen phosphorus. So there's a few parts of this graph that are important to look at when we're looking at recommendations. The first part they need is all soil fertility. People like zeros, so controls, so we know kind of what happens. So this is our yield at 0, which, for example, in this field was 41 bushels per acre. It's a drier field. Then we have our optimal yield, which is this part over here. That's 76 bushels per acre. And then from that, you can get what a lot of people call as relative yield, where you take that 41 divided by 76, you get 54% or 0.54. And then another aspect you can look at is that change in yield. So we're looking at the difference between our optimal yield, whatever phosphorus rate that was, versus our 0. And that can be defined as a change in yield or a delta yield. And for example, this is 76 minus 41 is 35. So then once you take those fields and you do lots of these all over the place, you can take that soil test level of 12 parts per million, that 54% relative yield, kind of make those lines in the middle. And that's where that one field goes. So this is why people always need lots of fields, because you have lots of places to do this with different soil test levels, so we can actually make that relationship there to get that critical value. And this is South Dakota data, but that's the concept there. And then same thing with phosphorus calibration. You take that 12 part per million in that example, we need 71 pounds of PTO5. and you're going to develop that relationship with lots of other fields. So that's kind of the calibration part of the context of what we've been working on. Some of that's basic, but some of that's stuff that sometimes we don't understand that's going behind our recommendation. So it's kind of a good idea to know how they're developed, so then we can use that information as we're making decisions on our own farms as well. So in evaluating the soil test level for South Dakota, I'd started there 6 years ago, and no one knew when the last time it had been revisited. So we did the work in a bunch of different fields throughout eastern central South Dakota to kind of look at that. And this is typically, so you saw what we'd like. We have a relative yield here. We have our Olsen phosphorus test here because we have higher pH soils. So this is typically the test that we use. And then we have our responses here. This is around our 100% line that we're trying to figure out where that kind of curves off. You can see as we're in lower yielding areas, and this is where art meets science. You got to figure out what line represents that area, or what represents those field areas to make a decision, because based on what model and what other things you choose, it can kind of change what decision you make. So trying to figuring out what best represents those areas. And in South Dakota was around about 15 to 20 parts per million, which was pretty similar to our current 16 threshold, where we'd say at Olsen 16, you typically don't see responses above that. So a lot of new data saying kind of similar to what we already know, which isn't a bad thing, helps us confirm that that recommendation is still fairly valid for our South Dakota soils. This is all soils together. We've also been looking at some differences in no-till and things like that. So this is kind of that. We're going to look at this in a couple different ways. So a lot of times we look at soil test phosphorus, we think of categories, we think very low, low, medium, high, very high, and kind of what those mean. We're looking at the same data just kind of as a different visual to kind of see it there. We can see our 0 to 5, or our very low soils. We don't have very many of them. They're below the 100%, are low. Here, our medium, then we start to hit that higher level. We're kind of getting closer to that 100% line. And then we get to that very high where we typically not apply fertilizer. You can start to see where they just kind of start to stay leveled out at right around that 100% line there. We're going to look at this last one. So now we're going to group them. This is your box and whisker plots for all you stats lovers. Those boxes, middle 50% of their data. The line is the mean and median. So we're trying to figure out where exactly that starts to go above 100%, just different ways to look at the same data. So this is our very low, our low and our medium, our high. So once we kind of get above this is where we start to hit that 100% line. There as we hit those soils. So again, it kind of shows us at those 16 to 20, once we start to hit 16, 20, we're right there on the 100% line. So typically we're not going to see a large ROI once we start to get there on some of these things, which is what we've seen in the past. There's obviously some variation because we're not perfectly there. Just to kind of show you, I think it's fun to look at different areas of the country to see how this looks in other areas. That's so northern part. So we're going to take a little travel down to Virginia, Maryland, North Carolina. This is with soybean. This is with the Malic 1 test. This is a regional or national project that we were working on, kind of looking at some of this data and some of the modeling. So here we're going to look at their response curve. You can see it starts out pretty low and goes up. They're very responsive to potassium for soybean, and you see it kind of represents and flattens out up here. And their critical value for Malic 1K is 57. We move to Arkansas still with soybean. Now we're going to the Malic 3 test that they use. So not quite as steep of a curve, but they still kind of had that same response pattern where they go up and then flatten out. Their critical value is 168. So you can see the differences just the soil tests can make and what those soil tests extract. So it's really important to have those relationships with yield and other components. So now we're going to go up here. This is Iowa, Minnesota, and South Dakota data. So you can see how much different some of the regional differences can look. A lot more compacted, but you can still see that same relationship there. You combine our data or look at just South Dakota's data, it's still around 16 parts per million Olsen. That they've found that critical value exists, or you no longer see a consistent response to phosphorus fertilization with these different tests. So there's several different ways you can look at a critical value. I'm just going to show some of them, some different ways. It's not just looking at relative yield. In this one, we're going to look at response frequency. So here, this is on our y-axis is our corn yield response frequency. So this is just if the field either increased in yield, so more than a 5 bushel increase, within 0 to 5 bushels, it was the same. If we had a negative yield, it decreased yield by 5 bushels. So those are kind of those categories that we made. So this is our soil test phosphorus. So we're first of all going to look at percentage of sites raised. So this is a typical pattern that you would expect. Lower soil test P means you had a higher probability you're going to see a response. We go from 80% down to 0 when close to 36 or 40. One thing that we don't think about sometimes, this is also spring applied phosphorus. Sometimes we don't think about, we can actually decrease yields in the first year sometimes, depending on how much phosphorus we're applying. So this is the percentage of sites that saw a lowering of yield when we added phosphorus. This was 100 pounds of phosphorus in the spring, so it was a higher rate. So if you don't apply that, just think about that too. We did see a decrease in yield. And then we also, very following the same trend, we saw no change in yield. So these kind of mirrored each other. Interesting thing is if you look at that intersection there, it's about 20 parts per million, which is right in that 16 to 20 kind of shown from the other graph as well. So it kind of shows us as we increase in soil P, we decrease our chance of response, but we also increase our chance of not seeing a response or seeing a decrease. So the next way, it's kind of sometimes a little bit less abstract. You're not thinking in percentages, you're not thinking in relative yields, but you're thinking in actual yields. So this graph looks at yield change. So if we remember this graph up here, the difference between the optimal yield and that 0 yield. Looking at that change in yield as we increase in soil test phosphorus here. So we have our 0 line right here. So we didn't change at all. So we saw kind of what you'd expect. As you had lower soil test phosphorus, you had a higher increase in yield change, around 15 to 20 bushels per acre. And as that soil test phosphorus increases, we see that yield change start to decrease as well. Hits a 0 around 22 parts per million. Another thing I like to say, if you want to see a 5 bushel per acre yield increase before you're doing something actively, so a 5 bushel per acre right here, drop that over, you go down to around 16. So it kind of shows us we can look at this data in 3 different ways. We're still finding that similar range of answers between about 16 and 20, depending on what information you want to gather together to help you make a decision of whether you're fertilizing or not in your field. So one other thing to think about, kind of putting all this data together, is we typically think of categories, and when we're making decisions, whether it's a low, medium, or high, or very high soil testing soil. So here we have our very low, low, medium, high, very high, and just kind of above that. So we're going to look at a few different things here. This is our first of all, those first ones we're looking at are responsiveness. So you can see we're higher responsive in the lower soil testing soils, but we're still down here, we're still responsive. We still saw yield change in about 40% of our 40 to 30%. But then the real thing about that is, yes, there was a response, but is it worth it economically? So then if we look here at our relative mean yield data, so anything close to 100% is, 100% is maximum yield. So here we see our 80s and 90s. And then once we get above that threshold, we're no longer typically fertilized, we're going to see we're really close to 100%. So not a whole lot of yield change. And we can see actual numbers here, where if we're below that line there, where we typically see fertilization is helpful, we're going to see, 16, 17, 13 bushels per acre increase, drop to 8. Then once we get above that, that's when we start to see, yes, we might see a percentage responsiveness like over here, but that median value of response is pretty low. I'm seeing that yield change. That's the median. This is just kind of that middle 50% of the data. So between 9 and 24 to 2 to 18. Then once we get here, we have some negative that we saw some responses. And then, we still had some that were up there. We're still trying to figure out some of those, and I'll be later in the presentation, but kind of see that the range and increase becomes much lower once we start to get above those values when we're adding phosphorus. So we're thinking about the economics of our fields and where we're spending our money. It kind of helps us. Yes, we might see a change in yield, but is that change in yield worth it to put the phosphorus down? So just in summary of some of the things that we've seen, just kind of gone over that critical value, depending on which way we slice it or do it, it was between 16 and 20 for our Olsen test. If we look at confidence intervals, 13 to 22, as far as some of those different models are showing us, which is what a lot of farmers, depending on what they're doing, are kind of working at keeping those in there, depending on what range of risk you like to take. If you want to see that 5 bushel or 5% increase, bound 16 parts per million is what we're showing there as well. So the big thing of, if we look at just using soil test phosphorus, we want to know how accurate am I in that? And across these sites, we have about 40 sites in this data set across eastern and central South Dakota. That's about 68% accurate. Just using soil test P, if I were to tell you, if you're giving me your soil test P value, if I said yes or no, and it'll respond, 68% of the time based on this data, I'll be right. So then the question, is that good enough? And if we can, how can we improve? So that's kind of some of the things we're working on with some of our data. So to kind of figure out, this is just soil test P, this is what your standard they've been doing since the 40s, 50s. What else can we do? So kind of trying to figure out what else can we do to improve this. I kind of took a step back trying to figure out, what are some things, looking at our landscape, it's really stepping back and looking at the soil functions that we know our soils are hydrologic buffer, their food, biodiversity, habitat, nutrient cycling, filtering and buffering. They provide physical stability. So there's all these things. So how can we enhance that? And how do these things affect our phosphorus cycle? And one of the quotes that one of my colleagues likes to use in some of his presentations I thought was interesting. It says, while the chemistry and physics of the soil system provides the context, it's the soil biota which is adaptive to changes in environmental circumstances. So if we kind of look here, we can kind of see that, our biology, they change based on the weather, based on the soil parameters that are in that field from year to year. We kind of set the stage. We're not going to change our soil texture in that field quickly, but those microbes and other things that are in there, they can adapt what's going on from year to year. So what can they potentially tell us? And if we talk to the NRCS, or just, looking at our soil health principles, and people have talked about several of these at different points today. If we think about how well we're armoring the soil, what residue we're keeping on the soil to reduce erosion, keep water going into the soil, what our disturbance looks like, what's our plant diversity, what crops are we planting and what rotation. Are we getting living roots on that system? How does that affect our phosphorus cycling? Are livestock integrated, or are they not? How does that affect some of those cycling and things like that? And so if we look at these soil health principles and see how they affect the soil, one of my colleagues, Hannah Poffenberger, put this slide together, which I really liked. We think about principles of soil health. How do they affect the soil? Well, we look at chemically. One thing that they do, they can help buffer our pH. They can help mineralize nutrients, and they can help, retain those cations on for the nutrients, so we keep them, and so they're not washed off or lost through erosion. We look at our physical parts of our soil. We want to improve greater aggregate stability. We want to look at water holding capacity and filtration. How are these soil health aspects affecting that as well, as far as our nutrient management, what we're measuring? And biologically, we want these systems to have a diverse, active microbial community. So then the question kind of becomes, if we're looking at soil health, we're looking at management, looking at crop growth, how does that influence this? And this is kind of a diagram that kind of, we're looking at what changing in management. We want to see that develop some soil health improvement, something we can measure and look at. And then because of that, we want to see increased productivity from better conditions in our field, whether that's moisture control, nutrient cycling, or things like that. And once we do that, once we see better conditions or see improvement, then we're likely going to want to keep doing something to improve our soils. And maybe that'll lead to different and more management changes and practices over time. So if we kind of look at what specific management changes that might be, that could be something regarding tillage, could be interseeding cover crops or planting them after, could be adding a crop to your rotation where you can economically do that. So there's lots of different things we can potentially do. There's just some ideas, but ultimately we're trying to figure out how to take these soil health principles and how we can use them to help improve our nutrient cycling and things like that. So one of the quotes one of my friends said, it says, data without a decision is just trivia. So we have a lot of data that we can get from our soil test reports. So we're trying to figure out how to use that data to help improve the decisions that we can make. And one of the biggest things, if we're making management decisions to help us keep those decisions, we need to be able to measure things over time. And a lot of times we're looking at those physical, chemical, and biological things. And there's lots of different soil health frameworks. We're just going to kind of go over a couple some results. This is from some results from Missouri, and it's a soil health assessment framework. There's some other ones out there that's going to show some there. But what this does is it takes scores from 4 different areas. It uses a physical, biological, nutrient score. You can see what they do here, a chemical score. So they did this in a bunch of different systems in Missouri. And we can kind of divide a line down here. These are your perennial systems. These are your annual cropping systems, which would kind of make sense. You know, the less you touch the soil, the more you're going to have some of these things. What we're going to focus on is these ones over here, because over here we have our cool and warm season grass pasture type of situations. Over here is our annual cropping systems. So what makes them different from each other? The major things like this are rotational diversity, no-till, mulch-till, things like that. So if we're able to kind of take these in more of a continuum, we see that our pastures, perennial systems are kind of on the higher end, but as we work here, interesting thing is our best option is when we incorporate no-till plus cover crop plus looking at our rotation. Adding all 3 of those together gave us those best soil health opportunities. So once we start taking them out, then you can see we lose cover crop, you use change your tillage type, you can kind of start going down the other way on that spectrum. So a lot of times when we group these things together, we see better responses over time. So kind of going back to this, looking at these fertilizer decisions, we want to see data. We want to see something behind them. So kind of an example of hopefully what we're wanting to see is we kind of look at this same type of thing here. We look at our yield, how it can change from low to high. We look at our nutrient input, and we've seen this before. Our typical response curve, what we see in soil fertility a lot. So we see as we increase in nutrient input, depending on there, at some point we're going to optimize and we're going to kind of plateau on that yield. The question is if I change my management practice, what happens to this area of the curve? Here's a couple of options that we're potentially looking for. That we could just increase our yield. We still needed the same amount of nutrients, but we increased our yield because we made that improvement in our management. Another option is we could move that nutrient. We maintain yield, but we maybe need a little bit less nutrients in there. Or the third option, which is what we're all hoping for, less nutrients and an increase in yield. So these are, to me, this is what, when I'm doing these different soil management, soil health aspects, this is what I'm looking for. What changes there from just this green line? Are we going up in yield and that's all? Or are we moving over less in nutrients or more, depending on what management practice that we're using on? So that's one thing to keep in mind. The other thing is nutrient uptake. So we're going to take a quick dive. We're going to go below the soil here in just a second. This is where I wish I had theme music for, keep on digging in that song. I should really find that one day to do this, this presentation to wake everyone up. But if we look at this, we see that below the root zone, we see our roots that are growing. We know that that's the interface of where those nutrients are being taken up, is right there. And if we look really deep into this, we're going to see that root and that soil water system reaching out for those nutrients. We have our soil aggregates that are in the soil. We have those air-filled pore spaces, and then we have our nutrients that are on those in the soil water or on the soil colloids. And we have our 3 basic areas that we're taking up nutrients through mass flow, diffusion, or root interception. So today's talk is on phosphorus uptake. And so that's mostly happening through diffusion. And so what that's doing is it's basically following an ion concentration gradient. So you need to go from a higher concentration to a lower concentration to get to that root. So once that starts getting that higher concentration, those nutrients are moving there. Once it kind of equals that concentration, then that nutrient slows down. So then the question becomes, this isn't the best process, it's not the fastest. How can we improve that? What about phosphorus do we need to know as far as it's being taken up that we can improve this right here? So one other thing to know is that arbuscular mycorrhizae help with phosphorus uptake. So they dissolve phosphorus, help with drought resistance, help with aggregate stability. And one thing that they've seen is they're higher in no-till areas because you're not constantly cutting them down. So what they're doing, just kind of to show, is they're associating with that root. They're reaching out there and they can help uptake that phosphorus that's farther away and help increase the amount of phosphorus that those roots can uptake into that plant and hopefully increase that availability for those roots. So this is just another diagram showing that same idea. This is where the plant can get to as far as phosphorus uptake, and then those arbuscular mycorrhizae can hopefully reach out and increase the access to phosphorus for that root. So with those 2 ideas kind of in mind, There's a project in central South Dakota working on looking at phosphorus, and specifically looking at these two areas. If we minimize disturbance, we increase our plant diversity, how does that influence our phosphorus fertilizer needs? So just giving a quick diagram of this, minimizing disturbance, back in 1990 or so, they switched from till to no-till. Plant diversity, they went from a corn-soybean rotation to a 5-year kind of a stacked rotation, soybean, small grains, cover crop, soybean, 2 years of corn. So this is central South Dakota, it's dry. They do have irrigation on this one, so keep that in mind. We have our crop rotation. So they added 3 levels of phosphorus, a low, medium, and high, a very high. So low and medium, we should see a response to phosphorus fertilization. The very high, we don't expect. So these are the yields for them. We're going to start in 2018. This project's been going on for about 20 years, but we're going to start in 2018. So our low is our red, our blue is our medium, and our yellow is our high. So you can see in 2018, regardless of that soil test level, very similar yields. 2019 wheat, very similar yields, all 3 soil test levels. 2020 soybean, very similar, all 3 test levels. 2021 corn, see a little bit more variability here, but ultimately statistically no difference. 2022, a little bit more variability, drier year. This one's technically a little bit lower, but not enough to economically fertilize with that low soil test level. So the question is we've seen this, we have different soil test levels, but we saw the same response to phosphorus. They still yielded similarly. So we wanted to know why. So this is where that mycorrhizal fungi comes into play. This is the number of propagules. It's just a measurement of mycorrhizae, so higher is better. So this is our low, our medium, and our high. So you can see pretty dramatically that low soil test level seems to have more mycorrhizae than that medium and high. And that trend is followed throughout the years. You can kind of see it dips down. These are drier years and wetter year. And overall, the average, you can see that the lower soil test P soil has more mycorrhizal fungi, which might be why they still yielded similarly. Doesn't show us that we can, what this kind of helped us kind of realize is potentially we can lower that soil test phosphorus needed in some of these systems. That critical value might go from 16 maybe to 11 or 12 to get that critical value instead. We haven't looked at those responses yet. It still needs phosphorus, but it doesn't need as high of a level to optimize yield for that one. So that's one project that we worked on. The other part of this is we remember this graph right here. Here we know that it's predicted 68% correctly as responsive or not responsive. So which ones are correct and which ones are not? So we want to figure out where our error is. So if we look at these right here, these ones are good, they're correct. That means they're below that critical value. We said they'd respond to fertilizer and they did. These ones, we say they're above that critical fertilizer. We say they wouldn't respond and they didn't. It's these ones right here and these ones right here that didn't behave like we want them to. There was something about them, so they were below. These ones right here are below that critical value, so we say they should need fertilizer, but they didn't. And these ones are the opposite. So we want to figure out how do we move these ones here and these ones here. What else do we need to know about the soil? So if we think about all the different chemical measurements, the physical measurements, the biological measurements that we can make, part of this project is wanting to know which of these is helpful, because typically we focus on these ones here, mostly just phosphorus as we traditionally do. What else do we need to know to help improve our ability to make good predictions? So this is just a quick study to show some logistics. We did this, shows 28 locations around central and eastern South Dakota. We've increased that since. Within each of these sites, we would take 3 to 5 places within a field that were hopefully different phosphorus levels. We'd have a control, then we'd add phosphorus, add potassium, add sulfur, and see what the response was. Within each of these 40 by 40 foot areas, we'd take a soil probe, get down kind of that horizons, texture, organic matter, bulk densities, measurements, and then we do some soil fertility as well as some soil health testing. So there are lots of different measurements. Just going to cut to the chase. What variables improved our phosphorus recommendations? So along with our Olsen phosphorus, we also had soil respiration, which is a measurement of microbial activity. So how much those microbes are respiring. So Technically, those ones that are respiring more, just like when you're running, you're respiring more higher levels of CO2, then that's measuring that. Our CEC, cation exchange capacity, as well as our clay content. Important thing to note about this is when we're looking at adding soil tests, we don't want to add a lot of money to our already very high soil testing bills. So a lot of things with this, Olsen P is what we typically use. CEC is not going to change a lot, but it's also part of a typical package. Clay content, great thing about that is once you kind of know your soil texture, you shouldn't have to redo it. The biggest one here is soil respiration, to seeing if it's worth it to add that too, because that's another $15, $20 test sometimes. How much that changes from year to year, which we're still working on. But those are some considerations. So with adding soil tests, you kind of have to have a framework. There's lots of different ways to statistically do this. We just kind of made a decision tree to help kind of see what it is about the soil we needed to know and why during some soil health events management practices, we weren't seeing responses when we should have. So the number we need to worry about is the top number. That's how many sites responded. The bottom number is just the percentage of sites in that box. So we start with 100% of our sites, 55% across the data set responded, so pretty close to 50-50. So once we add soil test phosphorus, if we're above that, which is where we typically say you don't respond. So here we want to know why they did respond when they weren't supposed to. So just using that, 32% of them responded. It also wanted us to look at cation exchange capacity. When we are above 19, it said still don't fertilize. It's not a very good chance. But when you are below 19, it's saying you still might have a chance of fertilization or see a response. So there's some of that there you can think of. If you're above that 17, you're a little worried about your field. So if we're looking at Olsen phosphorus and you're less than 17, this is where we typically say always fertilize, but we know some fields didn't respond. So just using that number, 61% responded, which is, what we typically think. So next thing I wanted to use was soil respiration. So it's saying if you have a high soil respiration, you have a still 40% chance of response, but also want to look at clay content. So if you had a low clay content, which is 55% clay is really high. So most soils are going to be below that, but you didn't have a very, only 42% chance of response. Whereas if you were high in clay, you had a much higher response. So if you're below 17 and you had high clay content, you're even more likely to respond to phosphorus fertilizer. So the other aspect of this is if you're below 124 in your soil respiration, so lower soil respiration, that's when you definitely had the highest chance. So low soil respiration, less microbial activity gives you a higher chance of needing to respond to phosphorus fertilization. So this is kind of framework, a little more complicated than using just soil test P, but we're working on testing it to see how accurate it is beyond the sites we've done so far. And then kind of looking at this, so what did we overall see? So we looked at, we just reminded ourselves that Olsen phosphorus by itself, 68% correct. How much did we improve? We went from 68 to 74%. Did it go up to 90? No. Did it improve? Yeah. So we're looking at some of those items to see if we can continue to further improve those items to see if we can, what else do we need to know about that soil so we can become, more accurate whether we're going to see a prediction or we're going to see a response to our phosphorus application or not. So that is what I have for you today. I guess I can take questions with whatever. Got 5 minutes. I did good. How does the pH level of the field influence the critical value in your research? I looked at pH because I know a lot of places use pH to differentiate their phosphorus. And it didn't come out. I could make it come out in some of the stuff I did, but it didn't come out easily. I think a lot of that might be because our soil tests are all typically a little bit higher. We don't have a lot of acidic soils. We're getting some on the eastern part of the states as we get more weathering. But we don't have a high of a range. Like when I was in Minnesota, we had a much higher range in soil pH, and I think was where they're able to differentiate those out. Where in South Dakota, in the fields that I had, I didn't have a wide enough pH difference. But we know phosphorus relates to pH because a high pH, it goes to calcium, and a low pH, it ties to iron. We didn't see enough of that in South Dakota soils to differentiate it, but we did, but I know it's there. Will AI be a tool for fertility recs in the future? Everything's going to AI right now, right? I think the more tools we have, the more information computers can process, more information than I can and can understand it. I showed you a diagram. There's also other models that it's just a black box kind of approach, which is hard for me to sit up here and present to you about why things happen the way they did, which is why I presented a diagram. But there's lots of different ways that AI is. The hard thing is it's not easily explainable because sometimes you don't know what's going on in the background. But I think they can process more information than I can, and there's more tools. So yeah, we're going to probably use more AI and more machine learning in the future because once you get beyond soil test phosphorus, it becomes much more complicated as far as interactions go. We all know that from our soil classes. So I assume AI is going to be able to get those and understand those better than we can to help us make our decisions. Higher level, do you think higher level AMF is directly related to low STP? And this one, yes, I think there's other factors. This is a long-term no-till field. It's irrigated, has a 5-year cropping rotation system. So I think all those are impacts. But this isn't the only research project that's seen different levels of soil test phosphorus affect AMF. So I think the different levels do affect it. Because if the plant doesn't need it, they're not going to send out the signals to get the AMF growing and saying, yep, I'll pay you the money for you to come do your job. They're not going to at higher soil test P levels, which is probably some of the factor there.
Other questions for Jason?
You looked at the economics behind any of this?
I'm working on it. I haven't. I first wanted to see if it worked. So that's why I said, doing the additional soil tests cost money. So we want to make sure that 68 to 74% or that 68 to 80% accuracy is worth the money that you'd be investing in those additional soil tests, which we all know and understand. So that's part of what we're working on is we got this concept done in the first few years. How accurate is it and is it worth it financially is kind of the next steps.
Other questions? Like we're good?
All right, thank you.
Thank you, Jason.